Hi everybody, welcome to our module on carbon dioxide. So recall that carbon dioxide is produced by cellular metabolism and it's transported to the lungs so the body can eliminate it when we exhale. And there are three mechanisms by which carbon dioxide gets to the lungs. The most important is in the form of bicarbonate. About 90% of the carbon dioxide is converted to bicarbonate and transported to the lungs. We'll talk about that in a minute. There's a smaller amount that's bound to hemoglobin. We'll also talk about this. And then finally, a tiny amount of carbon dioxide is just dissolved directly in the plasma. So let's talk about transport of carbon dioxide in the form of bicarbonate first. So carbon dioxide can be reacted with water and it can then be turned into carbonic acid and it can then be turned into bicarbonate and H+. And once carbon dioxide goes through this reaction, essentially the carbon dioxide has been stored in the form of bicarbonate here, which is how it's going to get transported to the lungs. The enzyme that catalyzes this entire reaction is carbonic anhydrase, and there's a lot of it found inside red blood cells. So that's going to catalyze the conversion of carbon dioxide and water into bicarb and hydrogen atoms so that the bicarbonate can carry the CO2 up to the lungs. Now, converting the carbon dioxide into bicarbonate has a couple of other effects that you need to know about. First of all, this reaction is going to produce a hydrogen ion, and it turns out that those hydrogen ions can bind to hemoglobin. And when a hydrogen ion is bound to hemoglobin, we often abbreviate it as HHB. It doesn't bind to the heme molecule the way oxygen does. It binds to the end of the globin chains, and it converts hemoglobin to a taut form, which releases more oxygen, and this shifts the oxygen curve to the right, and this is called the Bohr effect. It's easier to understand the Bohr effect if we look at a picture. So let's imagine that this is the oxygen dissociation curve of the body, and the more oxygen that's present by the x-axis down here, the more saturated hemoglobin is with oxygen. That should make sense to you. So let's suppose we're in the lungs, and there's a lot of oxygen in the lungs because we're breathing air. So we have a PO2 of about 100 in the lungs, and that corresponds with a point right about here, which means we are about 100% saturated in the lungs. Now let's suppose we go down to tissues where the PO2 is much lower. Say it's about 50 because tissues are consuming oxygen, so the PO2 is much lower. Well, if we were operating on this blue curve, we would now be at this point right about here, which means hemoglobin is about 75% saturated. So in going to the tissues, hemoglobin unloads oxygen such that it drops from 100% to 75% saturated. But the nice thing about the Bohr effect is that in addition to offloading this 25% like you see here, we shift the whole curve to the right. And that's this red line that I've drawn here. So now let's suppose we're at 50 right here. Now we shift up to this point on the red curve here. Well, that's a hemoglobin saturation of about 50. So because of the Bohr effect, instead of dropping just from 100 to 75, we drop from 100 all the way down to 50. So the Bohr effect helps hemoglobin unload additional oxygen, which helps the body get the oxygen that it needs. And the reason we have the Bohr effect is because of those hydrogen ions that are present, and the reason they're present is because we're converting carbon dioxide into bicarbonate and acid. So I've summarized the Bohr effect for you here. We have a rightward shift with more O2 offloaded for any given PO2. We operate in the blue curve when the level of H plus is low, like in the lungs, but we operate on the red curve when the level of H plus is high, like in the tissues. And that's the Bohr effect, and it's a consequence of the fact that we carry carbon dioxide in the form of bicarbonate and generate hydrogen atoms. Another consequence of carrying carbon dioxide in the blood in the form of bicarbonate is something called the chloride shift. So the red blood cells convert the carbon dioxide to bicarbonate, as I said before, via carbonic anhydrase. This is going to raise the level of bicarbonate inside the red blood cells, and remember that bicarbonate is negatively charged. So when the level of bicarbonate inside the cell gets high, it will diffuse out into the plasma. However, because the red cell is losing a negatively charged ion, it needs to gain some other negatively charged ion in order to maintain electrical neutrality, and the ion that it's going to gain is chloride. So chloride enters the cell to maintain electrical neutrality, and the result is that red blood cells have a very high chloride content, especially in the venous blood. So on the venous side, red blood cells have picked up a lot of CO2. I've shown that down here in this picture. It's been converted to bicarbonate and acid, and then the bicarbonate diffuses out of the red cell and chloride enters, and this keeps the amount of positive and negatively charged ions inside the red cell equal, and that's called the chloride shift. So everything I've been discussing up until now has been about carbon dioxide transported in the form of bicarbonate, but now I'm going to talk about carbon dioxide transported bound to hemoglobin. And when hemoglobin binds carbon dioxide, we call that carbaminohemoglobin. And it turns out that in a low oxygen environment, hemoglobin is more likely to bind CO2, and this is called the Haldane effect. And let me explain this to you so you understand why it's helpful to the body. So just like with the Bohr effect, the Haldane effect is easier to understand if we look at a graphical illustration of the principle and walk through it slowly. So in this picture, I've drawn the level of carbon dioxide in the blood on the x-axis here, and the amount of carbon dioxide bound to hemoglobin on the y-axis. And it follows a relationship like this purple line here, such that the more CO2 there is in the blood, the more is bound to hemoglobin. So let's start off by imagining we're in an environment where there's relatively low carbon dioxide, and the lungs are an environment where there's relatively low CO2 compared to tissues. If that were the case, we'd be operating on this purple line right about here, which corresponds to a relatively low amount of carbon dioxide bound to hemoglobin. Now let's imagine we move to an environment where there's high CO2, like in the tissues. There's a lot of cellular metabolism, a lot of CO2 being produced. That would move us to a point on this purple line right here. So we would shift up the purple line to this point right here, and that corresponds to a higher amount of hemoglobin bound to carbon dioxide. So if we had to stay on this purple line, when we move into tissues, we would increase the amount of carbon dioxide bound to hemoglobin by about this much. However, just like with the Bohr effect, there is something called the Haldane effect which shifts this curve. When you go to an environment like tissues where there's relatively low oxygen, you shift to the blue line that I've drawn right here. So now let's imagine again that we're in that high CO2 environment like the tissues. Well, instead of stopping at the purple line, we can move all the way up here on this blue line, which corresponds to a much higher level of hemoglobin saturation with carbon dioxide. The result is that we are able to bind much more carbon dioxide to hemoglobin in a low oxygen environment like the tissues, and this is because of the Haldane effect. And the reverse is also true. When you go to the lungs, you are able to offload much more CO2 because the curve shifts from the blue back to the purple. So I've summarized the Haldane effect for you here in this picture. When we're in an environment like the lungs where there's a high amount of O2, we operate on this purple curve here. But when we go to an environment where there's low oxygen, like in the tissues, we shift to the blue curve over here. And the result of the Haldane effect is that we're able to bind more carbon dioxide in the tissues, and we're able to unload more carbon dioxide in the lungs. So let me just summarize for you some of the differences between the tissues and the lungs in terms of respiratory physiology. In the tissues, you have a relatively low amount of oxygen because oxygen is being consumed by metabolism. You also have a high amount of carbon dioxide because it's being produced by metabolism. The carbon dioxide results in formation of H+, and this is going to favor oxygen unloading via the Bohr effect. And you can just remember that the O in the Bohr effect stands for O2, and that's why it's related to oxygen unloading. Also in tissues, you have conditions favorable for CO2 loading, and that's via the Haldane effect. And just think about the tissues and what they want to do. They want to unload oxygen and they want to load CO2, and that's why the Bohr effect and the Haldane effect promote these things. In the lungs, you have a relatively high amount of oxygen coming from the air and a relatively low amount of CO2 coming in the air, and you're also exhaling and letting the CO2 in the bloodstream leave the lungs. You have a relatively low concentration of H+ because the CO2 is low, and conditions in the lungs favor O2 loading via the Bohr effect, and they favor CO2 unloading via the Haldane effect. And again, you can memorize this if you want, but really all you have to do is think about what the lungs would want to do. Naturally, they want to load oxygen and unload CO2, and so the Bohr and Haldane effects work to make this happen. So what I'm going to do in the rest of this module is talk about a couple special situations where carbon dioxide plays an important role. These are high yield for your board. So we'll talk about carbon dioxide in high altitude and in exercise, and then we'll talk about how carbon dioxide affects cerebral blood flow and the control of respiration. So if you climb a mountain, say a very tall mountain like Mount Everest, the atmospheric pressure is very low up there. This means that you're going to have a low PO2. And because of the low PO2, your body's going to sense the hypoxia and stimulate hyperventilation. When you hyperventilate, you blow off more CO2, and this creates a respiratory alkalosis. So always remember that a respiratory alkalosis and a high pH are the expected findings in someone living at high altitude. Now, after about 24 to 48 hours, the kidneys are going to increase the amount of bicarb that they excrete, and this is going to make the pH fall back down towards normal. So up here on the right side of the screen, I've summarized the findings you would see in a person living at high altitude. They would have a high pH, they would have a low PO2, they would have a low PCO2, and they would have a low bicarb. And all these are normal responses of the body to the hyperventilation that's caused by low PO2 and low atmospheric pressure. Other things that happen at high altitude is that you will have the stimulation of erythropoietin from the chronic hypoxia. This is going to raise the hematocrit and hemoglobin over 10 to 14 days. So patients living at high altitude can have a mild or even moderate polycythemia, and this is from the increased EPO level. Another thing that rarely occurs but can happen is that you can get chronic hypoxic vasoconstriction. So recall that the vessels in the lungs vasoconstrict to hypoxia. This is a mechanism to shunt blood away from underventilated portions of the lung. Well, if the entire lung vasoconstricts from low oxygen levels, you can get pulmonary hypertension, and over time this can lead to right ventricular hypertrophy, and it can even lead to right heart failure, which is called cor pulmonale. You should also be aware of the effects of high altitude on the oxygen-hemoglobin dissociation curve. So there are two effects here that are important. The first is the hyperventilation is going to cause alkalosis, which shifts the curve to the left. This is sort of a reverse of the Bohr effect. We'll look at a picture of that in a minute. But there's a second important effect, and that's that the hypoxemia stimulates the red blood cells to produce 2,3-BPG. BPG is bisphosphoglyceric acid, and it's normally present in red blood cells, and it promotes the release of oxygen. And in the setting of high altitude, more 2,3-BPG gets produced, and this is going to shift the curve to the right, which means it will move back towards normal compared to the effect of alkalosis. So let's take a look at a picture here. So suppose that the blue line here is the normal oxygen hemoglobin dissociation curve. Well, the alkalosis is going to tend to shift it to the left, like to this green dashed curve I've shown right here. However, the higher levels of 2,3-BPG are going to shift it to the right, like this red line here. So the cumulative effect is that you're going to end up right back where you started from as a blue line in the middle. The two effects largely cancel each other out. Now let's talk about what happens to carbon dioxide when you exercise. So when you exercise, your body begins to consume more oxygen as the muscles do more work. The increased metabolic activity creates more carbon dioxide, and this is going to trigger an increase in ventilation. And as you know, if you go jog around the block, you'll start to breathe faster. That's a natural consequence of exercise. So let's talk about that in a little more detail. So when you exercise, more carbon dioxide is produced by your muscles. This means that the CO2 levels in the venous blood are going to rise, not the arterial blood. That stays normal. We'll talk about that in a minute. In addition, your muscles are going to consume more oxygen. This means the oxygen level in the venous blood is going to fall. Again, we're talking about the venous system, not the arterial system. The O2 level in the arterial system stays normal. We'll talk about that in a second. So on the arterial side, the increased ventilation and blood flow is allowing you to maintain a normal PaO2 and PaCO2 despite the metabolic changes. So very importantly, exercise should not cause you to become hypoxic, nor should it cause you to retain carbon dioxide. If your body is healthy, you will increase your ventilation rate and the blood flow, and you will take care of that extra carbon dioxide. You will be able to exhale it, and you will bring in more oxygen to meet the demands of the body. So I've summarized this for you down here because they like to ask you this on test. In the veins, the O2 level falls and the CO2 rises. That's a consequence of extra metabolism. In the arteries, the O2 and CO2 levels stay normal. You will on occasion see patients with cardiac disease or pulmonary disease who have their oxygen level fall when they exert themselves. This is abnormal, and this is not just a consequence of the exercise itself. It's because their heart and lungs are not healthy enough to meet the demands required of their body when they exercise. So let's talk about what this would look like in a picture. So on the x-axis here, I've drawn the length along a capillary in the lungs. On the y-axis, I've shown the partial pressure of oxygen. And at rest, you follow a curve like this red line I've drawn here. At rest, the blood is moving relatively slowly through the lungs, so the oxygen level rises rapidly as it moves along the capillary and quickly reaches the maximum amount and then levels out. With exercise, your blood is flowing much faster. This means that it gets a lot further along the capillary before it reaches the maximum value. However, notice that it does reach the same normal maximum value just like at rest, and that's why you don't become hypoxic with exercise. This is a fairly classic graph, so make sure you understand it for your boards. Now let's talk about carbon dioxide and cerebral blood flow. So carbon dioxide is a powerful cerebral vasodilator, and it's the main determinant of the amount of blood flow to the brain. So let's look down here at these two curves. This one on the left is for oxygen and this one on the right is for carbon dioxide. So the normal range of oxygen is shown right here. And you can see that when your oxygen level goes up and down within the normal range, it really doesn't change the cerebral blood flow over here on the y-axis. Basically, oxygen doesn't affect cerebral blood flow much at all unless it gets very, very low. If your oxygen level gets very low like down here, then it does begin to vasodilate the cerebral arteries and increase blood flow to the brain. But within the normal range of CO2, you can see that the CO2 level has a powerful effect on cerebral blood flow. As your CO2 level goes up, then the brain thinks it wants more blood flow, and so therefore it raises the blood flow. So these two curves are pretty classic. Make sure you understand their shape and why they're different. And the bottom line is that under normal circumstances, carbon dioxide is what determines how much blood flow there is to your brain. Oxygen really only has an effect if you get severely hypoxic. Under those circumstances, the cerebral blood flow will go up. So they like to ask you about clinical correlations of these physiologic principles on your boards, and one place that the cerebral blood flow and carbon dioxide levels have a physiologic correlation is in panic attacks. So people who have a panic attack will begin to hyperventilate, and that means that the CO2 level in their bloodstream will get low. This is called hypocapnia, and this will stimulate cerebral vasoconstriction because, as I just showed you, CO2 levels affect cerebral blood flow. It's thought that this contributes to the CNS symptoms of panic attacks like dizziness and blurred vision. Now, in reality, the symptoms of panic attacks are complex and not completely understood, so this is just one component. But it is important to know that the panic attack will lower the CO2 level and that can lead to vasoconstriction in the brain. Finally, let's talk about carbon dioxide and control of your respiratory rate. It's important for you to know that the level of carbon dioxide in the bloodstream is the major stimulus for breathing. It's not oxygen. The place that responds to the level of CO2 is in the medulla. There are central chemoreceptors in the medulla, and they tell you how fast to breathe based on the carbon dioxide level. There are also some peripheral chemoreceptors in the carotid and aortic bodies. These can sense CO2, but they're more sensitive to oxygen. And just like with the brain, they only kick in when you get severely hypoxemic. Under normal conditions, it's your central chemoreceptors in the medulla that are telling you how fast to breathe. And if they sense a high level of CO2, they will increase the respiratory rate. And if they sense a low level of CO2, they will decrease the respiratory rate. There are a couple of pathologic states where this CO2 control of respiratory rate is important, and one of them is in COPD. So COPD patients chronically retain carbon dioxide. They always have a high level of carbon dioxide in their blood. And the result is that over time, their response to CO2 can become blunted. When this occurs, the oxygen level becomes the major stimulus for breathing. Remember, I said it's sensed by those peripheral chemoreceptors. So something that can sometimes occur is that you can have a COPD patient and you give them 100% oxygen for dyspnea, and this can actually cause respiratory depression. What's happened is the hypoxemia was stimulating their respiratory rate, and you took that away when you applied oxygen, and this lowered their respiratory rate. Now, in reality, we give oxygen to COPD patients all the time, and it's safe to do, but you do have to monitor them closely, and you don't want to give too much because that can depress their respiratory rate. When this happens, the CO2 level will get very high, and they'll start to get confusion related to the high carbon dioxide level. Another clinical scenario where carbon dioxide and breathing control is important is in determining the ventilatory status of the patient. So when patients come into the hospital with lung disease, oxygen is often administered, and this raises the O2 level, but they can still be hypoventilating, and the only way you're going to know that is to check the carbon dioxide level. So essentially, the oxygen can mask the hypoventilation. It will make the oxygen saturation be 95 or 96 or 97%. It'll look normal, but the only way you're going to know that they're underventilating is to check their CO2 level. So this often happens in patients admitted with pneumonia or sometimes COPD or asthma. They'll be given O2 via nasal cannula. This will make the oxygen level normal, but if you check the blood gas and find that the CO2 level is high, for example 60 is high because normal is 40, this means they are hypoventilating, and this is often a patient who's going to need to be put on a mechanical ventilator because if not, the CO2 level will continue to climb. Another clinical scenario where the CO2 is useful to monitor ventilatory status is in patients who have neuromuscular diseases like ALS. So if you give oxygen to these patients, you'll raise their oxygen level to the normal range, but on a blood gas, if they have advanced ALS, you'll find that their CO2 is high, like 60, and this means that they're hypoventilating. Their respiratory muscles are failing because of the neuromuscular disease. If you don't treat this, these patients will start to get symptoms of high CO2. They'll get lethargic and confused and agitated, and eventually they can go into a coma and die, and that's why patients whose CO2 level is climbing usually need to be put on mechanical ventilation. And that concludes our module on carbon dioxide.